Moving on with the proceedings of the conference, we have Oration 5, Dr. N.S. Dhalla Oration, and Sir is here with us. A very good morning, Sir. Sir, good morning. Dhalla, sir. Yeah. So I'm going to introduce about the oration. Dr. N.S. Dhalla Oration is a prestigious honor established to celebrate the remarkable contribution of Dr. Narendra S. Dhalla, an internationally renowned cardiovascular pharmacologist and pioneer in cardiac function, pathophysiology, and therapeutics. This honor recognizes exceptional scientists for their exemplary work in biomedical research, especially in cardiovascular science, pharmacology, and translational medicine. The oration offers a platform for the awardee to articulate impactful research finding or transformative advancement relevant to clinical practice. The oration not only commemorates Dr. Dhalla's extraordinary legacy as a researcher, mentor, and leader, but also inspires the scientific community to pursue innovation and deliver meaningful contribution to human health. Chairing this session, we have two individuals who really exemplify academic excellence and mentorship. We have with us Vaike Gupta sir, and I think a stalwart in the field. He's being already introduced to you all. And second, we have Dr. Shiv Prakash sir, who's a distinguished leader in pharmacology and drug discovery, notable for founding the Synchron Research Services. May I request both of them to please come and take a seat on the dais? Can we have Gupta sir? I think he'll be joining soon. Yeah. So may I request you to introduce the speaker?
Good morning. Good morning to all of you. This is one of the most prestigious oration, and it makes it more special. Professor Dhalla himself is in the first row. Thank you very much, sir, for your contribution for IPS for instituting this prestigious award oration. I'm really honored to introduce today's speaker for this oration, Dr. Tejal Gandhi. Dr. Tejal Ricky Gandhi is professor and principal at Anand Pharmacy College, Gujarat, and associate dean at Gujarat Technological University. With over 33 years of academic and research experience, she has guided 150 plus postgraduates and 13 PhD scholars, published over 200 research papers, and holds 20 patents, a fellow of IACS and IPA. She has received numerous honors, including the AICTE Vishweshwaraya Best Teacher Award in 2020 and Distinguished Leadership Award IACS 2016. She also serves as Vice President, IACS India Section, and President, APTI Gujarat State Branch. I request Dr. Tejal Gandhi to present a talk, and it is my honor too that Dr. Tejal and I shared the same lab while we were doing PhD together at LM College of Pharmacy, Gujarat University.
नहीं नहीं चाहिए please. I'll 100% finish in 30 minutes, but to be, yeah, I'll keep in mind. Very good morning, esteemed chairpersons, respected pharmacologists, and dear delegates. Today I stand here profoundly grateful and deeply humbled for this recognition. First of all, thanks to this jury members for nominating and considering my nomination for this prestigious award that is named after my revered mentor Professor Naranjan Dalla, who continuously inspires and empowers countless scientists across the world. And special thanks to Dr. Prakash Diwan, because he nominated me for this oration, and jury members also for the same. I am really happy that this 1.5 day, this organizers, they took lot of care, and the scientific sessions are also wonderful. So thanks to organizer, and special thanks to Anil Kumar for this wonderful organization of this 55th IPSCON. So before going for this journey of this Betulin, I'll just give brief about this. In 1990, I went into this, we can say, lab of Professor R.K. Goel and Dr. D.D. Santani. We were doing this M.Pharm. I did my M.Pharm, and at that time, I just worked on this working heart preparation at that time, and I was so lucky that this during M.Pharm only I kept this in a poster presentation in Indian Pharmacological Society annual conference, and I got my life's first best poster award from IPS only. Followed by that this Kofik prize, and I continued my research and PhD also on this the compound that are acting this newer compound that was developed in LM College of Pharmacy, and we worked on this rabbit and dog, and that finally that compound this Cadila Pharma was very much interested for formulation development. So we have worked for this couple of years under the guidance of this Dr. Goel, and we published, and I got this S.C. Verma Memorial Prize also in this IPS Gujarat chapter. So this my journey continued, and then in 2002 we can say that the first time I thought that why we cannot give this evidence to this natural product, and I got this grant from Gujarat government, and I tried for this cabbage juice, and we got this very good results for that. This followed by, I can say this, by this slides I will talk about that also, but today I will focus more on this my last 3 years, this that is the one compound we are working on that. So this the topic of this my oration, that is decoding the cardioprotective symphony of Betulin from transcriptomic signature to therapeutic this promise. So this topic will have this we can say intersection or embodies this computational biology as well as the experimental biology, and which will be really helping us to find out this how this natural compound having a promise to become a cardioprotective this compound. As we all know that this various reports and everyone knows that in news also this people are suddenly the death cardiac death in young people are increased, as well as we can say not only that, but the children young as 8 also collapsed due to this sudden cardiac death. And people may speculate that this is post COVID, but ICMR study had proven that there is no link, and if we will really go into the depth of this epidemiology, this tragic death is increasing significantly since so many years. So let us have a look very quick at this epidemiology of the last decade. What happened this in last decade, that so many countries, European and that countries, this significant decrease in this CVD death were observed, but however in India we can see this the 15% rise in this cardiac death were observed, and particularly this, as we all know that in male it was higher as compared to this female. The interestingly you can see this out of this death, this 50% approximately death is due to this ischemic heart disease, and the most this modifiable 4 risk factors remain the unchanged the 10 years back and till today. So this really we can say that this next is, as everyone knows, that 17.9 million deaths were reported by this WHO every year due to the CVD diseases. And in India, this recently we can say in 2024, one study said that this 31% total death is due to the cardiovascular disease in India also. And one study, this 2-3 years big study that that report this indicated that in India approximately 90 million Indians are living with cardiovascular conditions. But the most alarming thing is that the cardiovascular death rate of 272 per 1 lakh population in India is much higher than the global average 235. And particularly in that study they have mentioned this, particularly the rise in this West Bengal and Punjab is quite high. Not only that, this in my state, or we can say this in Gujarat also, in 2023 this rise in this emergency call of cardiac death 2108 is significantly increased. You can see that from 31% to 42% approximately. So, this is really alarming, and that really needs this, we can say that the awareness as well as the further research. So, if we will go into this very quickly, this in this decrease, or we can say this ischemia that lead to this oxidative stress, ROS and inflammation, reactive oxygen species formation, hypoxia and inflammation, that may lead to this. Ultimately there are cascade of events happen, but we can say this decrease this availability of oxygen at mitochondria that lead to this disruption of ATP production, and finally this pro-apoptic BAX and caspases are upregulated. To maintain this thing, that we can say this various this cardiac cell, they will try to cope up this situation, and this beta receptor activation, they will try to this cope up with the contractile dysfunctions, and this angiotensin receptors are trying to maintain the blood pressure. But the overactivation of this may lead to this ROS and PI3-AKT pathway stimulation or activation, and angiotensin that may lead to this MAP kinase and ERK pathway. This inflammation that leads to this toll-like receptors, and that ultimately we can say this further complicating this heart response to this type of stress. And as we know this, there are various stages and that are interconnected, that different pathological stages are there, and they all are regulated by different signal molecular pathways. This when we are talking about this any time for the cardioprotection and functional restoration, this multi-targeted approach is really that helping us to modulating the cell signaling. And here you can see that in different pathological state, that hypertrophy, fibrosis, inflammation, various pathways are involved. Interestingly, this pathway are not working in isolation, they are making one network, that is the coordinated network, and that regulating this cardiac injury and remodeling. So, it is very much important that any compound that is having this type of crosstalk between this pathway, that may be beneficial for this, we can say this cardioprotective agent. So, if we know this various synthetic drugs are having this, of course, it is widely used, but besides that, this toxicity profile, or we can say this withdrawal from the marketing, and that may lead to this nothing but this rise in the problem. And in opposite to that, this interestingly this natural compounds, that's this in silico toxicity, experimental toxicity data that shows this very good result. And not only that, but in clinical trials also, that they are the success we can see in this graph. So, with this we can say that this normally, as you know, this in a natural drug discovery program, people used to in India that they were extracting and then doing this crude extract in vitro or in vivo, and sometime we can say we are going for a pure isolate. The problem with the natural product scientists is that the scale up, and the scale up is the major problem. So, nowadays we can say, so in our laboratories also this as I have mentioned that we have started in 2004. After that, we have also started evaluating this various crude extracts from the plant as well as this active constituent for this. But during this time, that active constituent, like as for example, we have worked on the pomegranate juice as well as this punicogelic acid, that is the active constituent. And surprisingly, in genetic expression, we found that the pure juice was giving very good effect. But the problem is that when we are giving this, the quality and this nothing but this markers, and that is quite difficult. So we have done this so many plant evaluation in various, this is only I am reporting that are only that we have evaluated in this cardiovascular diseases, and that may lead to this various patent also in this from our laboratory. So with this we can say that we decided to work on this. Nowadays this natural scientists, as I have mentioned, that they are working using this CAT technology, and in which that they are going for this number of library of natural compounds, they are exploring against this various disease target. So we also in our this work in last 3 years, so I will focus this work in detail, this what we have done particularly, that we can say this molecular docking study, and with this we have use this traditional knowledge and ethnopharmacological activity that reported, and from that we have tried to select this few natural compounds. With that, this drug likeliness prediction in silico study also we have done, and pharmacokinetic and toxicological prediction that we have done. For the shortlisting the natural compound, we wanted to take it forward for the cardioprotection. Here you can see that this traditional use of this various compounds that we have mentioned, and here you can see this various ethnopharmacological activities that is already reported. So from this literature work we have shortlisted 5 compounds: betulin, betulinic acid, quercetin, lupeol and lycopene for our further study. So after that, and this while we have done this, the in-depth this all compounds are acting on a different this molecular pathway, and this that multi-targeted this potential will be beneficial for this, particularly this after oxidative stress, this cascade of events are happening, that it may be beneficial. So with this objective we have started this. So now first for this any compound that we wanted to take it further for oral administration, this Lipinski rule is the very much important this for the prediction, and any compound having a more than one violation of this Lipinski rule. I am not going into the depth of this Lipinski rule, we have done this with the software, and here you can see this lycopene is violating these 2 rules, and because of that we have skipped this, we have not taken it further for the investigation. So with this, then finally this 4 natural compounds we have docked again this various with the dock site platform protein plus server, we have checked with the different this active sites, and finally this we have docked against this various targets like a state tree, where you can see that 3D representation, and then we found this very good binding affinity with this betulin and quercetin here. Not only this dock score was good, glide score was also good, and besides that that gives a very good precision. Besides that, inhibition constants are low and number of H bonds are higher, this hydrophobic interactions are more. That shows that the stability in between this ligand and this protein interaction. And similarly we have done with this NF kappa B also, all these 4 were docked, and we also found this similar result with the betulin and quercetin. Then we have done with this ATP2A1 this ligand complex, and here also we found good result with this binding affinity is good with this betulin, quercetin and betulinic acid, and but with this JAK2 we have not found this that much good result, only quercetin and betulinic acid showed this good effect. So this it is very much important that this docking and this, but the prediction of this ADME, that is very much important if we wanted to take it further. So for this finalization of the compound, that we have taken this ADME, this software we have used, and because here you can see that this profile of this ADME is quite good, but this blood-brain barrier permeability, that is the limiting factor, because that may predict the risk of CNS toxicity, and HERG liability assessment is really this, we can say this, giving idea about the cardiac toxicity. So HERG liabilities is quite good, so it is a safe cardiac safe compounds, but here only this quercetin is having an excellent this blood-brain barrier permeability, and because of that you can see that there is a risk of this CNS toxicity. This the another this prediction that we have done this the ProTox2, that is the toxicity profiling, and here you can see that this lupeol, quercetin and betulic acid, this all three compounds, they are this natural compounds are showing this some toxicity endpoints, and as compared to this the betulin is showing this very good toxicity profile, having a LD50, that is the prediction of this 2000 milligram per kg LD50, and acute toxicity class, that is GHS, it is class 4. So, with this we can say this docking result and this in silico study, that is a part 1 of this project, and that we found that quercetin having a very good binding affinity with this various target, but this if we will talk about this toxicity profile, we have this not taken it further for this our investigation. So, we selected this betulin having this favorable binding affinity as well as the good toxicity profiling and this ADME parameters. So, we have taken it further for this our research work. So, in this part 2 we can see this betulin. What betulin is? It is a pentacyclic triterpenoid, and it is a widely this abundant amount is present in the birch tree, and it is this it is present in the bark of the birch tree. Recently this review article also showed that this is acting, because I am focusing on the multi-targeted this targets in this cascade of events of this oxidative stress, or we can say this in the cardiac injury. So, this they have mentioned also anti-inflammatory and antioxidative activities, and the acute toxicity report actually this on this mice, they found that it is class 6, means it is having a very good toxicity profile also. So, this with this for this cardiac injury we have used this isoprenaline induced I think some problem with this pointer. So, this we have divided this animals rat in this 5 groups: normal control group, model control group, that is isoprenaline is given at the 2 doses of 85 milligram per kg subcutaneously at the interval of 24 hours, and we have used standard demivolol for this and given 28 days treatment. This group 4 and 5 we have given this betulin 20 milligram per kg and 40 milligram per kg respectively for 28 days, and besides that definitely in group 3, 4 and 5 we have given this iso administration like a model control group. After that we have this analyzed various parameters. So, very quickly this part 2 this preclinical study, that we can have an idea about this results of this betulin, and the heart function was this we can say this cardiac protective effect we can see from this key parameter that are restored, like a mean arterial pressures, left ventricular end diastolic pressure, positive dP by dt, negative dP by dt, and that shows that betulins this cardioprotective this activity. Besides that this ECG, that in that this we have found that this ST significant elevation with this iso administration, which was protected by this betulin dosing, and this notably ST elevation was decreased, P wave and QRS complex also, that positive results were there. If you see this infarct area, that is significantly reduced with the treatment of this betulin. So, we have again this cardioprotective this activity that we have evaluated this by this cardiac markers, and then you can see that this betulin is also this decreasing this significantly decrease the CKMB, LDH, troponin levels as well as the calcium. So, with this we have also seen the expiration of the inflammation, and interestingly over here in a 40 milligram per kg this betulin dose showed a very good this results, particularly with this inflammation, and again this anti-apoptic so this caspase and that, so this betulin activity, it significantly reduced the expression of the caspase as well as this BAX also. So, this betulin is reported as this preventing this oxidative stress, and we also found the similar results. You can see this MDA levels are significantly reduced, that is lipid peroxidation is decreasing, and this activity of this antioxidant enzymes that are also significantly increased with this Betulin treatment. This histology study that also showed that this isoprenaline lead to this infiltration of this, we can say that accumulation of inflammatory cells, this necrosis, edema, and this is reduced in this standard drug as well as Betulin. But significantly this restoration of the architecture we found in a 40 milligram per kg dose of this thing. And from this result, we decided to take it further. This left ventricular, we have this tissues that of this left ventricular of this normal control, model control, and this Betulin 40 milligram per kg that we have undergone for the transcriptomic study. Here you know that just checking this genes on which Betulin is working or not, instead of that we have done this transcriptomic analysis that will give idea about this whole transcriptome sequence, what is happening at that level, and particularly genome. So, in this we have extracted this RNA, and RNA having a more than 6.5 this ring, we have this taken it further for this library preparation. In library preparation we have used this 2SQ standard total RNA platform, and then we have done this depletion, fragmentation, ligation, and finally that we can say we went for this sequencing on the Illumina Novasec 6000 platform. We generated this FASTQ files, and reads were mapped against the red genome using the STAR software, and we have done the quantification of this differential expressed gene by this feature counts, and the important thing, this analysis of this differentially expressed gene were done using the DESQ2. This biological interpretation of this differentially expressed genes are very much important. We have used this KEGG analysis, Synego, gene ontology for this particularly for transcriptomic analysis. The first we will talk about this isoprenaline. What happened with this isoprenaline administration in this particularly transcriptomic analysis. 1445 genes are differentially expressed in the left ventricular heart because of the iso administration. And if we will talk about this, the geoontology. This geoanalysis is nothing but it is a post-genomic tool, and that gives this biological meaning to each gene, and that is very much important. Here you can see that this various terms that are enriched. Here you can see that this are enriched, and here you can see this is nothing but this p values and this FTR. So, this from this we can hypothesize that this, yes, in isoprenaline this geo terms in a we are clubbing and we are interpreting, we have interpreted that this isoprenaline insult may lead to this nothing but this metabolic this metabolic drive, as well as we can say this hypoxia, oxidative stress, apoptic cardiomyocyte death, as well as nothing but this cytokine surge was also there. This, and next is that is we can talk about this the remodeling, also proliferation and fibrosis. So, this gene ontology can help us to this. I am not going into the depth of this, that data I am not presenting, but we have done for the cellular components analysis, we have done the biological process analysis, we have done the molecular function analysis, and all this will give us idea about this how this isoprenaline is actually doing this at the gene level changes or differential expression of genes. So, the next after geoontology, it is very much important that 1445 genes are differentially expressed. Out of that, this definitely this some are this we can say this significantly expressed. So, what happened is these genes we had given this biological meaning, but what happened at this we can say this at pathway level. As we know that in the pathophysiology of this cardiac injury various pathways are involved. So, we have done this KEGG analysis, and KEGG analysis gives the idea about that this different pathways that are enriched because of this isoprenaline administration. And here you can see this MAP kinase signaling pathway, cytokine receptor pathway, PI3-AKT pathway, and this node-like receptor pathways that are enriched, and number of genes are this. So, if I will keep into the tabular form, in KEGG analysis you can see that these are the signaling pathways that are this p values you can see that significantly enriched, and the number of genes in each this pathway because of isoprenaline administration, how many genes are affected. I am not going into the list of that, but this many number of genes are. But if I will just give you a pictorial this thing presentation, this different pathways you can see this red color that are differentially expressed genes. So, in this MAP kinase signaling pathway, PI3-AKT signaling pathway, we have analyzed this JEXTAT pathway, apoptosis signaling pathway and necroptosis signaling pathway to understand that which type of differentially expressed genes are significantly altered because of isoprenaline administration. So, this, but the important thing, yes, we understood that thing, which are the pathways enriched with the isoprenaline, which are the various genes are differentially expressed, but what happened with the Betulin treatment. So, here you can see the gene expression analysis, and here the left side you can see this the Venn diagram, the 650 genes are this differentially expressed and common in this iso as well as Betulin treatment. So, this isoprenaline administration leads to this differentially expression of 650 genes. That 650 genes out of 145, 1445, this 650 are again this normalized by this Betulin. So, this, and this is interesting result. Here you can see that this out of that 650, 366 are down regulated, this upregulated with the iso, and which is normalized or down regulated by Betulin treatment. And at this molecular level you can see this, the 284 are this down regulated by isoprenaline administration that may lead to this upregulated by B. So, this the visually we can see this how this B is effective. So, this is the scattered plot, volcano plot, and here you can see this the red color that are upregulated genes, green color are downregulated genes. The left side that is normal versus iso, you can see that various genes that we have this in this, but we are not going into the depth that this is nothing but this B versus iso. In that this volcano plot, if you see this the x axis that is a narrower, and so you can see this the volcano plot is narrower and the wings are shorter, and that reflects that this genes moves towards center, and so this regulation or we can say this expression is decreased, and we can say this or increased because negative and positive is there, and fewer significant genes because the wings become shorter. So, this is also showed that in this scattered plot we can see that this Betulin is showing a very good effect on this genes or we can say pathways in which they are involved. So, this KEGG analysis or we can say geo-ental analysis, what happened with this Betulin. So, we have shortlisted out of that this prominent genes which are involved in the cardiac injury, and this genes you can see that are this isoprenaline is changing their expression. Out of this 12 genes, 10 were upregulated by iso and 2 were downregulated by this isoprenaline. So, this Betulin this, and here you can see the first 3 TLR7, NLRP3 and OS2 is related with everyone knows about with the inflammation. Then EGR1, IRF9 in June that is related with the cardiomyocyte death, and next is that is MMP16 and COL3A1 that is important for this cardiac remodeling, and this LRRN4 that is important for this we can say this related with this ER stress this response or modulator. CNP-1 is everyone knows that in significant in clinics also this is the significant role in the cardiac repair, and this ADMTS2 that is a fibrotic mediator, and CCN1 is the early diagnostic marker. So, this all are significantly normalized by this, and in the last column you can see that this in geontology we have again categorized all them with this, whether it is involved in the this cellular components or this biological processes or molecular this function. So, now this again KEGG enrichment analysis, these genes they are differentially expressed, they are this protector, or we can say this restoration happened with this Betulin treatment, in which pathway they are involved. So, here I have kept this the first 3 significantly effect this altered this genes with the Betulin. So, you can see this cytokine receptor enriched interaction pathway, ERB signaling pathway, MAP kinase signaling pathway, various genes are there, not signaling pathway, and necroptosis and JAK-STAT pathway. In all this thing that this various this PI3 AKT pathway, this which are the genes that are altered and that are again this we can say this Betulin have a positive effect, or we can say this normalize this alteration or expression. So, this TGB signaling pathway. So, we have this many pathways are enriched, and in that again this various genes are dysregulated genes are normalized. So, then finally, that we can say this expressed genes, this individually we are talking about the genes and their biological, or you can say their involvement in the pathological path means various signaling pathways. We have undergone for this PPI, this protein-protein interaction analysis using the cytoscape, and this protein-protein analysis will give you idea about this at this differentially expressed genes, which proteins are this expressed, and the interaction between these proteins, because that is at the molecular level interesting. And here you can see that this EGR1, that is a stress we can say this stress related this EGR1 protein, as well as this another is that is June also, as I have said that this correlated with this cardiomyocyte death. There are, so from this we can identify this hub proteins, which are very much important for the this we can say this cardiac injury, and whether it is change or there is a change in the expression of this protein or not. That is the one thing that we have evaluated, and second is that is interaction. The more this interaction means the crosstalk is more. So, if we are able to this, we can see that this here, this if we are able to like CCN1, EGR1, and there is a this immunity related, this innate immune related OS2 is there. So, that collaboration is that is again making a whole inflammation related this network, and if we are able to this address this, there is a possibility that we can address this multi-target that are involved, or multiple pathways involved in the cardiac injury. So, here you can see this again this down regulation of this, here this MSLN, this LRRN4 and interleukin 1,4, that again talks about this cardiac repairing and this inflammation. So, this is and again this side you can see this Betulin, that is this affecting this hub proteins and restoring this disturbed network. And so, again from this result also we can say that this Betulin is showing this cardioprotective effect. The interesting thing is that it is at the lab level. There should be a correlation with this what is happening at the clinical level. So, what we have done that we have analyzed that this myocardial infarction patients, what are the genes that are altered. So, these are the list of this genes that are altered in the myocardial infarct patients tissue, that, and these are the reported. And interestingly, out of this many genes that I have talked about, these are the genes that are again quite similar, that with the ISO we can see that this administration that may lead to this dysregulation. And interestingly, that we found that this very good correlation, that this all genes are again this normalized with the Betulin treatment. So, this is really proving that this possibility of this we can say this translation of our this research from preclinical to computational to this we can say go for this clinical studies also. So, in summary I can say that this yeah in this sorry, but this is not reported data, but this is the interesting new finding that this is never reported this with isoprenaline. These are the genes that are yellow highlighted, that are first time we have reported, that are this in transcriptomic study we found that this genes are differentially expressed and clinically correlated also. So, still we have to publish this data. In summary, I can say that this in silico foundation, this Betulin showed a strong docking with inflammatory, this apoptotic targets supported by favorable this prediction of this ADME and this toxicity profile, and that justify our selection of this Betulin for this cardioprotective lead. We have done the preclinical evaluation and we have seen that this oxidative stress, inflammation, apoptosis and restored cardiac histoarchitecture confirming the potent biological activity. The third that we have done the transcriptomic precision, isoprenaline dysregulate 1445 genes and activating pathways of stress, inflammation, apoptosis, necrosis and fibrosis. Betulin normalized 650 this dysregulated genes and corrected key GO and CAC pathways including MAP kinase, PI3AKT, JAK-STAT, apoptosis and TGF-beta. Of course, this PPI, that interaction, protein-protein interaction analysis identify the central hub, that is EGR1, OS2, CCN1, MSLN, interleukin 1A, LRR and 4 as drivers of inflammatory and remodeling pathways, and Betulin corrected this disrupted network, highlighting its multi-target cardioprotective action. And overlap with the clinical MI gene signature and reversal of the dysregulation by Betulin highlights its therapeutic potential for myocardial injury. So, in conclusion, we can say this is the journey of 3 years of this Betulin, that Betulin orchestrates a comprehensive cardioprotective response spanning molecular docking, biological efficacy, transcriptomic precision and network rewiring, positioning it as a potent multi-target therapeutic candidate for myocardial injury and future MI management. There are a lot of data. I have tried to keep few data from the transcriptomics. Thanks to Government of Gujarat for funding this the research and Anand Pharmacy College, and thanks to my mentor for this project, Dr. Chaitanya Joshi, Director of Gujarat Biotechnology Research Center, and Dr. Anita Mehta, this professor at our institute, and these two researchers, that Hital Shah, Associate Professor, and Tejal Shah from Gujarat Biotechnological Research Center. They have actually done all this research work. So, Ek Kadam Atmanirbhar Bharat ke our selection of this phytoconstituent is a tribute to traditional wisdom validated through preclinical, modern transcriptomic, docking and the science of pharmacokinetics. Thank you so much.
Thank you Tejas. I must compliment you for 2-3 things. One is extraordinary work which you have done, from in silico, then you did preclinical, and the most important thing is that you found correlation with the clinical MI cases. The young MI is still a mystery. Several projects by ICMR, DBT are being still in process, and registry is also there about young MI. How the incidence of young MI has increased 3-fold in last 20 years, 3-fold, rather 3.2-fold, which is the registry says, and still the exact cause is not known, multifactorial. But what you have found is the correlation with the gene and possibly a protective agent also, which will be a real. So that is first compliment to you. The second compliment is you finished much before your allotted time. So 3 minutes, I think you saved 3 minutes for us. So big hand for that.
I promised them I'll finish.
Great. So many leaders, they promise but they don't deliver. You promised and then you delivered. And the third biggest compliment is that you have delivered this oration in front of a person who made this oration, Dr. Dhalla. I think it's a very rare occasion that an orator gives an oration in front of a person who dedicated his life, his entire love to Indian Pharmacological Society, Dr. Dhalla. I think those young generation who are not fully aware of Dr. Dhalla's contribution to Indian pharmacology, rather I would say the cardiology in the globe is indebted to Dr. Dhalla, because he started the experimental pharmacology and correlation with the experimental pharmacology and clinical correlates in the globe. He is the first Indian to reach such heights in not only cardiovascular but entire experimental and cardiovascular. I visited his city as his guest, and it was such an honor feeling for an Indian to see Dr. Dhalla's bust in one of the most prestigious park, where there are busts of 10 or 11 people who have been given Order of Canada award. And I think at this age, sir, you are so passionate about Indian Pharmacology Society that at this age, coming from Canada, staying here, and I saw him yesterday also full day attending, and today also I was just talking to Shiv, that I think heads off to you. So big salute to him. I think we give a standing ovation to Dr. Dhalla as well as Tejas to you. I think that's a great, sir. So, and I wish, Dr. Dhalla, you come again and again. I was told by Shiv that perhaps you showed reluctance that I may not be able to come next year, but I am sure that you will come. So, thank you very much. And because this is an appreciation of your hard work, so we don't ask questions, but we only clap for you. Thank you. Over to Shiv.
I think we should conclude this quickly because we are running out of time. Please.
Thank you so much, ma'am, for such an informative session, and thank you to the chairs. Now is the time to felicitate our speaker and the chairpersons. I request the chairperson to kindly honor our speaker with a token of gratitude.